Hi everyone, we'll be talking to you today about cacti distribution in Sonderman Park. So first, we'll introduce our first specimen, Opuntia, or more commonly the prickly pear cactus. The prickly pear is characterized by its long spikes and cladodes, the multiple lobes that serve a similar function as leaves. They grow anywhere from southern Canada to Mexico. As with all cacti, the prickly pear plant is a cam plant, meaning that it opens its stomata at night to intake carbon dioxide and then converts it into a carbon compound to store until daylight, when it can use that carbon dioxide to synthesize it into sugars. They're moderately drought tolerant. Lastly, the prickly pear reproduce in two key ways. They flower and therefore spread through seed dispersal, but they can also reproduce when their cladodes break off and reroot. So our second specimen is the hedgehog species. The technical name for this is Echinocerius, but we refer to it as the nylon hedgehog. It's very similar to the prickly pear cactus, but a key difference is that the hedgehog cactus is incredibly frost and drought tolerant and has roots that are up to 10 to 20 feet deep. While the hedgehog also reproduces sexually and asexually, the asexual reproduction is through root colonization, which from my understanding is kind of like aspen suckers.
So what exactly determines species distribution? There are two main drivers behind species distribution patterns: exogenous and endogenous forces. Exogenous forces are at play when something else out there has made a pattern of distribution, either patchy or uniform, and then the species at hand is just tracking that distribution. For example, this could be caused by other organisms such as gophers or voles that dig up dirt, and then the cactuses will grow on that exposed soil, following the rodent's pattern of disturbance. Endogenous forces are driven by interactions such as facilitation and competition, both inter- and intraspecifically. For example, imagine a species with lots of intraspecific competition. You wouldn't see that species clumped. Instead, the distribution would be fairly evenly spaced out like a checkerboard, since each individual wants to be as far away as possible from other members of the population. In this study, we wanted to look at the distribution of nylon hedgehog cacti and prickly pear cacti, both within each species and between the two species.
Hi, welcome everybody to our study area. Here we have our nice area of interest. It's a square defined by all of our personal belongings.
So once again, we have our two species, prickly pear cacti and the nylon hedgehog. To observe the distribution of these two species, we marked out an area of interest that was 15 by 15 feet that included both species of cacti. Then, we split the area of interest in half. On one side, we looked at both the prickly pear and the hedgehog cacti, and on the other side, we only looked at the hedgehog cacti. To collect data, we used a GPS unit to mark the coordinates of each cacti, while taking notes on the species type and the number of cladodes or units of the individual. Here's an example of how we collected our data points.
We have been eyeing this beautiful patch of nylon hedgehogs.
We counted 23 little units in total, so we're going to do a data point with 23 nylon hedgehogs. Pretty exciting. After collecting and uploading the data, this is a map of the distribution we got. The larger the dot, the more cladodes or units there were on that cactus, which can be a proxy for size or age. The pink dots are prickly pears, and the blue dots are nylon hedgehogs. To analyze this, we used a statistical test called a G-function. The best way to understand this is to imagine that each data point has a circle around it. We can count the number of individuals that a cacti interacts with that are within the area of that circle, and that number changes as we increase the radius of the circle. To analyze this, we need a baseline. Imagine that a species were completely randomly distributed, and so the number of species inside the circle would increase proportionally to the circle's radius. This baseline of a random distribution is represented by the dotted yellow line on the graph. To illustrate this, here is a random hedgehog cactus from our area of interest. When we start with a small circle, there are no other individuals within the circle. Therefore, we fall below the line of random distribution on the graph. As we increase the circle, we encounter the many individuals clustered around it. Based on the graph to the right, this corresponds with a steep increase in slope where the species tend to be more clustered rather than randomly distributed, and we are now above the line of random distribution. As we increase the circle further, we zoom out of the cluster and encounter relatively fewer individuals. Looking at the graph again, this corresponds to the line dipping below the baseline random distribution, showing that at the larger scale, the species tend toward avoidance.
This figure shows the actual outcomes of the G function in our area of interest. Once again, we have the radius of the circle on the x-axis and the number of plants encountered on the y-axis. The first column, labeled 1, represents the hedgehog species, and the column labeled 2 represents the prickly pear. The blue line shows the random distribution, like we talked about previously. And the black line shows what was recorded in our area of interest. If you're above the blue line, the distribution is clustered, and if you're below the blue line, the distribution is over-dispersed. It's also worth noting that it's possible to have a pattern that's clustered at one scale and over-dispersed at another scale. In conclusion, these graphs show that for nylon hedgehog species, at a small spatial scale, described by a small radius, there's tight interspecies clustering, but this goes away at large scales. This is shown on the graph in the upper left. For the prickly pears on the lower right, initially the black line is hugging the blue line, so it means that they're fairly randomly distributed. But within a larger radii, the black line rises above the blue line, meaning that there's clustering, but only at a larger scale. The last two graphs represent the interspecies interactions. Both graphs show that at a larger scale, both species are avoiding each other. This next graph uses a similar conceptual framework as we just talked about with the circles. The difference here is that the y-axis describes conditional probability of encountering individuals within a certain radius, either of the same or different species. The green line here represents a random distribution, where the probability of encountering individuals remains constant as we increase the size of the circle. For hedgehog-hedgehog interactions, which is on the graph in the upper left-hand corner, a small initial dip below the green line represents a bit of interspecies avoidance. This, however, quickly changes, signified by a shift above the green line, indicating that on small to moderate scales, the species have a high probability of clustering. At larger scales, however, it will be more likely that the hedgehog cacti distribute in accordance to avoidance. For the prickly pear cactus, in the bottom right-hand corner, it's likely that on a small scale there's interspecies avoidance, while at medium to large scale there's a high probability of clustering. The graphs on the upper right and lower left-hand corners show the interactions between the two species. They show similar initial clustering of both species at small scales, and then avoidance.
In conclusion, for the nylon hedgehog, there's likely some kind of intraspecies facilitation at small scales that promotes growth. At larger scales, however, there must either be exogenous limitations or endogenous competition interactions that prevent clustering. For the prickly pear, there's likely competition for a single resource that prevents them from clumping at a small scale. Prickly pears are known to not be the best competitors, so this resource could be light or space, for example. The other option is that exogenous forces are at play. At greater scales, there is facilitation among the species that causes clustering. This animation shows the scaled intraspecies distributions of both the prickly pear and the nylon hedgehog. In terms of addressing both species, we see some facilitation at small scales and a prominent avoidance pattern at large scales. From the endogenous perspective, this could arise through competition and colonization. Exogenously, these results could be accounted for by disruption. This animation shows the scaled interspecies distributions for the prickly pear and the hedgehog cacti. Thanks for listening.